Hello, thank you for the introduction and for the invitation. Topic today is fluidic absorption drives cystogenesis in a human organoid on-chip model of polycystic kidney disease. This is a new paper that will hopefully come out from my laboratory, and it's really the first time I'm presenting it, and I'm excited to show you guys how it works. These are my disclosures. I'll start with the big picture here, and this is a different type of barrier from what we've been discussing. This is the kidney, and the kidney is a highly fluidic organ. It maybe is the most advanced microfluidic device on the planet. And these structures, these nephrons that create this filtration and then huge reabsorption of 180 liters per day, these structures arise only during embryonic development. So if you want to study the nephron and study the filtration processes that are involved, you really have to go back to the start before any of the organs have formed. And this is what we've been doing in the laboratory and many laboratories around the world, using human pluripotent stem cells to derive organoids, and then using the kidney organoids to study different disease states in the laboratory, and ultimately potentially study regeneration of the kidneys. So this is the protocol that I devised when I was a postdoc. Yes, it's good to do postdocs. This is how innovation happens. So it took me many years, but eventually I stumbled across a mechanism to generate human kidney organoids for the first time from pluripotent stem cells. And these are the types of structures that we get out of these differentiation cultures. There are complex podocytes, which are the filtering cells of the kidneys, attached to proximal tubular cells and distal tubular cells beneath those in a beautifully patterned distal to proximal axis. And these types of cells we haven't been able to grow before, we haven't been able to culture before in primary cultures, and certainly we've never achieved this type of organization before. So it raises a lot of possibilities. But for me, what I wanted to do here was really to study the potential of these cells to model disease. And not just any disease, but a genetic disease state called polycystic kidney disease, which has been a major challenge to model at the cellular level for decades. And the fundamental feature of this disease is that these tiny tubular structures within the kidneys expand, they balloon out, and they form these cyst structures that eventually crowd out the healthy tissue in the kidneys and cause kidney failure. Now, we've known the genes that cause this disorder for 25 years going on now, PKD1 and PKD2, but fundamentally, we don't know exactly what they do. They seem to encode a channel receptor complex that localizes to this antenna-like structure called the primary cilium at the apical surface of the cells. But why this results in cystogenesis has long been a mystery. And one of the great challenges, as I alluded to, is that cell lines from patients don't form cysts in a PKD-specific way. They may balloon out and form some spheroid structures, but those types of structures are also found in wild-type cells that don't have any disease state. So fundamentally, we don't have a reconstitution of this disease state in cell culture. Well, until recently, it turns out that our kidney organoids can beautifully reconstitute this fundamental genotype-phenotype correlation that's observed in polycystic kidney disease. And these are some of the images shown here. On the left, you can see the non-PKD organoids, and on the right, the isogenic gene-edited PKD1 or PKD2 knockout organoids. And these knockout organoids specifically form these large cyst structures that are absorbing fluid and expand over time in the dish. So this is really PKD in a dish. Now, most recently, we've studied this cilium, which is this antenna structure that I alluded to, where these PKD proteins are localized. And we've essentially knocked out this tiny organelle, the cilium, in human pluripotent stem cells. This is the type of experiment you really can't even do in a patient because it would kill the patient. But with human pluripotent stem cells, you can get away with a lot. And in this case, the cells are just fine without cilia. They can differentiate into organoids, albeit at a lower rate. But what's really remarkable is that once these structures form organoids, they go on to form cysts that are exactly like the cysts we see when we knock out the PKD disease genes. And you can see how large one of these organoids can grow in this movie here. This is a flask, and it's got an organoid that's gained so much fluid that is about the size of a ping pong ball. So we recently published that, and I won't go into it any further, just to say that we can see that there is a mechanism here specific to the disease state that's reproducible in different models. So the question that we wanted to get at next was, what was the effect of flow in this system? All these organoids, they've been generated under static conditions. We don't know how perfusion would affect cystogenesis. So to get at this, we actually arrived at a very simple system where we take our PKD organoids or our control organoids, and we place them in a very simple microfluidic device, and we flow media over the tops of these structures for a series of hours. And when we do this, what we observe is that the PKD organoids actually swell over time. You can see this swelling effect here. The cysts expand as they encounter the flow. And we don't see this type of swelling in the control organoid. So it seems to be something specific to the PKD. So we wanted to see whether this was really flow or some other component of the system. That's the issue here. To get at that, if we can go back a slide, to get at that, we had to generate a diffusive static condition. Initially, we just tried to plug in essentially a pipe, a syringe needle into our device and call that the static condition. Well, it turns out that that doesn't work because no diffusion is actually coming through the syringe needle into these channels. So that's not really a static condition, guys. But what we were able to engineer was a true diffusive static condition where we used a wide bore syringe and connected that to our device. And in this case, we're able to see the food dye diffuse into the channel. So now we know we're getting diffusion, and we can use a large reservoir, attach it to our channel, and do the same experiment with similar volumes, but without the flow. And when we do that, I keep pressing ahead. When we do that, what we observe is that volume alone partially substitutes for flow in the cyst expansion phenotype. And you can see these data quantified here, where at the left, the pure static condition where there's no diffusion, well, those cysts don't grow at all. They're not very happy. But if you use a diffusive static condition of 1 ml, it doesn't make a difference. But if you use 25 ml, you suddenly see an increase in the size of the cysts, which approaches the increase that's observed in the fluidic condition. It doesn't completely reach it, but it gets close. And moreover, you can eliminate the channel device entirely and simply place these organoids, as you see on the right, in dishes of different size and see the same effect, where over time, the organoids in the larger volume expand without any flow. So volume is important here. So what is it in the volume that is affecting the cysts? Glucose is an obvious candidate. We know that the kidneys, one of their primary functions is to absorb glucose. And we can beautifully see glucose absorption in these chips over time. This is one of the best things about these microfluidic devices. They're so thin that you can take live movies and watch the glucose enter the organoids and enter the cysts. And when we do this, we can see glucose being absorbed into the cyst lining epithelium and subsequently filling up the cyst. And this occurs in both the fluidic as well as the diffusive static condition. And there is a lag time in the diffusive static, but it reaches about the same levels of glucose absorption over time. So we know that the cysts are actively absorbing glucose, as is the rest of the organoid, the cells within the organoid. We also know that there are transporters of glucose that are specifically expressed at the apical surface of the tubular structures in the organoids as well as the cysts. So this then led us to the question of whether we could simply inhibit glucose transport and block cyst formation. And indeed, what we observed is that when we added in glucose to higher levels, we saw increased cyst growth. And when we removed or when we blocked glucose absorption using Floritin, which is essentially a broad spectrum inhibitor of glucose transport, we could dramatically reduce cyst growth. So this was about a 70% reduction in the expansion rate of these cysts. It actually went below the baseline. And we know that these levels of Floritin are not damaging the organoids because we perform live dead analysis as well as other types of metrics to look at the toxicity of these drugs before we do these experiments. And these are non-toxic doses. So simply inhibiting glucose transport blocks cyst formation. Well, this is all well and good, but it's a little bit confusing because in the body, the absorptive part of the cyst is facing inwards, and glucose transport would be leaving the cyst rather than filling it up. So we asked the question, what is the orientation? What is the polarity of our organoids? Is it like the cysts in the body, or is it different? And when we looked at the organoids, looking at acetylated alpha tubulin as a marker of cilia, as well as ZO1 as markers of tight junctions, well, we observed that there are tubular structures in the organoids that are properly polarized, as we would expect, with the apical surface lining the inside of the tubule. But the cysts, without exception, all face the other way, where the apical surface of these cysts, as you see in the upper left here, is facing outwards towards the media. So this is an inverted cyst. It is the opposite of the orientation of a cyst in vivo. And yet, it's capable of reproducing the phenotype that you see in vivo down to a T. So how are these forming? Well, we did closer analyses of these cysts, and you can see in the upper right here, it's really, the cyst is forming from a peripheral epithelial layer that almost surrounds the organoid. And those tubules that we're used to seeing are actually involutions of the membrane, of this peripheral membrane in these cyst structures. So that explains to us somewhat how you can get these tubular structures, but also have these peripheral cysts. And we took a step further and we took movies of these organoids as they underwent cystogenesis. And you can see this, one of these in time-lapse images at the bottom here. And if you look at that little yellow arrowhead, that's pointing out one of these involutions of the peripheral membrane. And then what you'll observe is that the peripheral membrane expands, the part of the organoid epithelium that's facing outward. It expands out very rapidly and forms the cyst, while that internal piece, which is the part of the cyst, part of the organoid that's polarized inwards, does not expand and remains inside this expanding cyst. So it's really consistent with the idea of an absorptive process coming through the apical side of the organoid epithelium that is driving cystogenesis in this system. Okay, well, now we had to think about how does this dovetail with what's happening in vivo, where the cysts have the opposite polarity. So we went back to a mouse model of polycystic kidney disease and took a close look at how the cysts were forming. And what we observed is that there were little pockets within these early cystic kidneys that were not lined by epithelium at all. Instead, they were pockets of fluid accumulation in the interstitium. And we could see this also by immunofluorescence at later stages. When you look at the cysts, there were areas where the cystic epithelium, which is, normally the kidney epithelium is very tightly associated with the interstitium and the endothelium. In this case, it seemed to be peeling away as if it was absorbing fluid and that fluid was pooling in the interstitium. Moreover, when we injected glucose into animals with PKD, we could see that this glucose was rapidly absorbed through the cyst lining epithelium and into the interstitium of the organ. And this gives us the idea that really what might be happening is that the cysts are absorbing fluid, and the problem is that the fluid is accumulating within the kidney, and this creates space within the kidney for the tubules to deform and fill out the cyst. So my major conclusion here is that, as the old saying goes, all models are wrong, but some are useful. In our case, our model has an inverted epithelium, but that inverted epithelium, which normally isn't there in nature, is actually driving cystogenesis through a reabsorptive process. And this teaches us that, absorption could be playing a critical role in PKD, and this has not been examined for many years. In fact, the focus has been on the opposite. The focus has been on secretion as a driver of PKD. But maybe we've got it wrong. Maybe the organoids are teaching us how the disease actually works, even though they're not perfect. So to sum up, organoids reconstitute PKD cyst formation in a disease-specific way. organoids on chips really drove us to this revelation that cysts form via fluidic absorption, and inhibitors of glucose transport are novel targets for PKD therapy. So I'd like to thank the folks in the lab. This has really been a multi-year project, and particularly Sienna Lee and Nellie Cruz and Louisa Helms have been drivers of this work. I'd also like to thank NCATS particularly, as well as NIDDK and the Laura Nowak Macklin Fund for supporting this innovative area of research in our laboratory. And I'll thank you for your attention.
That was brilliant, thank you. It's good to see a real use of kidney organoids. Just a quick question. Did you try staining them for the podocyte markers? Do you know how they organize?
Oh yeah, I mean, the podocytes are beautiful in the organoids. And in the cyst structures, the podocytes actually hang out on the exterior. So they're connected to the cyst epithelium. And they also polarize outwards towards the media, just like the cystic epithelium. But they do not really contribute to the cyst. It's the tubular epithelial cells and the distal tubular epithelial cells and the parietal epithelial cells. Those are what's giving rise to the actual cyst, which makes sense because those are the more absorptive cell types.
Yeah, great, thank you. Have we got any more questions? No, one.
Hi, thanks so much for the talk. I was wondering, you've presented on glucose data. Have you looked at any other small molecule metabolites or things that could be in interstitial fluid like electrolytes? And kind of follow-up question to that, have you guys investigated doing any kind of computational transport models and applying that to the data you've collected?
Yeah, both great thoughts. We haven't yet looked at any other small molecules, but yeah, it could be that there's something else driving the process, right? I mean, ultimately, it's got to be water that's flowing into these things and filling them up. We know that the cysts can exclude certain things from the media as well. So they're pumping in the opposite direction, and that may be co-transporter activity of some kind, which is driving the process. So now that we believe it's really import that's driving the cyst, I think it raises a lot of possibilities for what we could look at. So we haven't looked at those yet, but I hope to, and I would enjoy discussing over a beer maybe how to do that. Yeah, the second question in terms of computational modeling is also a very good one. We haven't gone down that road yet, but it would be interesting. I mean, one possibility is that the fundamental mechanism here is the pliability of the epithelium itself. In other words, the transport levels may be the same, but the membrane itself of the epithelium is weaker and allows for distension. So that would be a cool thing to try to model computationally.
Great. Thank you.
Thank you.
Nice talk. Thank you very much. When we grow organoid, we grow in matrigel. So in this kidney organoids, do you grow them in the matrigel? And also, I'm wondering about the effect of glucose, whether it increases osmolality and therefore it draw the fluid into the cyst. And I'm growing organoid, typically tumor organoid, and sometimes we see this cyst formation. And I'm wondering, is it kind of organoid phenomenon, or is it because of the PKD mutations?
All right, so first question, in terms of how we culture the organoids, they're actually cultured on a really thin layer, a thin matrigel sandwich. So it's just about 1% matrigel on either side initially. And that's in an adherent culture. And then we pop them off those plates into suspension cultures before we put them back inside the channels. And the channels are also coated with a thin layer of matrigel. So we use a thin layer of matrigel, but not a lot of matrigel. They're not baked in it. The second question, in terms of whether glucose, the osmolarity is actually driving the fluid uptake, yes, I think that's how it seems to be working. At least that must be the effect of these glucose inhibitors, is that they're blocking glucose uptake, and by virtue of that, they're blocking the fluid flow, which would be flowing in to balance the glucose on the other side of the epithelium. And the third question, you see some cysts in other organoids. I think cysts are a bit of a default configuration for a lot of epithelial cells. So, you know, the fact that a cyst is there doesn't mean it's PKD. But if you wanted to test and see whether it's PKD, then the right experiment is to take isogenic lines, control versus PKD knockout, and see whether you see an enhanced cyst formation under those circumstances. I'll say that like human pluripotent stem cells also express the same polycystin genes. They also have cilia. They also form cysts. But there's absolutely no difference in cystogenesis in the undifferentiated human pluripotent stem cell, whether or not you have the PKD gene there or not. So just because you have the mutations doesn't mean you're going to get a PKD cyst, and just because you have a cyst doesn't mean it's PKD. It really depends on being able to see the cysts specifically in the PKD genotype in the tissue of interest. But it does affect a lot of other tissues besides just kidneys.